Hello and welcome to another episode of the Prof series. In this episode, we'll cover papers made by the two main soybean breeders at the University of Guelph, Dr. Milad Eskandari and Dr. Istvan Rajcan. Dr. Istvan Rajcan teaches plant breeding and was the coordinator of the project I was working on that became the Soybean Experiment documentary I published in 2022. I had Dr. Milad Eskandari in the corn and soybean course in Rajcan. Both were very impressive individuals with a long pedigree of soybean breeding. Dr. Milad Eskandari is the soybean breeder at Rajcan, and Dr. Istvan Rajcan is the soybean breeder at Guelph. These two together have an almost monopoly on food quality soybean breeding, for simple reasons that their work is quite high quality in nature. We'll cover two papers by these researchers. Let's get started. The first paper was written by Dr. Eskandari and Dr. Rajcan in 2019, where it mentions that these two breeding programs have evolved quite a bit. The breeding program started in 1874 with the release of OEC 211, which was bred by Dr. Charles Zavitz. Since then, several high-quality and high-producing cultivars have been made, and most of the new cultivars since then have been bred by crossbreeding high yield to high yield. However, one of the consequences of this crossing scheme has been a narrowed genetic base, although there's evidence that genetic diversity has been maintained through the efforts of various breeders prior to Dr. Rajcan and Dr. Eskandari. But the researchers did not know for certain. So with the advent of genome sequencing technology, Dr. Rajcan and Dr. Eskandari compared 296 cultivars and lines, consisting of 69 elite cultivars from University of Guelph, 50 from the Rajcan campus, 43 historical cultivars, 53 University of Guelph experimental lines, and 33 Rajcan experimental lines. The genetic diversity of these populations were compared. Here are the results. Through the pedigree records, the 43 historical cultivars were released between 1907 and 1985. The elite cultivars have been released between 2002 and 2016. The results showed that the historical cultivars had the highest level of genetic diversity within this general group. Genetic diversity is lowest, however, in two locations. The cultivars released between 1950 and 1979, and 1990 to 1999. The second most genetically diverse group was the cultivars released between 1980 and 1989. And the third most genetically diverse groups were the cultivars released between 2000 and 2009, and 2010 to 2016. These modern groups, within the early to late 2000s and the early to late 2010s, showed a gradual increase in genetic diversity. It, however, works a lot differently within Rajcan. The cultivars bred between the year 2010 and current year are the most genetically diverse group within the Rajcan campus cultivars, with the lowest genetic diversity occurring between 2000 and 2009. Indeed, based on the breeding work covered using this genetic data, the first 20 years of breeding in Rajcan led to a decrease in genetic diversity, before increasing since 2010. Within the realm of the University of Guelph main campus, the cultivars had a similar trend, although with some slight deviations. The highest level of genetic diversity was found in the cultivars released between 1983 and 1989, again in terms of modern cultivars, declining in the 90s and further decreasing in the early 2000s to its lowest point, before a significant increase occurred in 2010 to 2016. The GM-GIA gene has been the critical gene of choice for breeding season soybeans. The breeding program to make soybeans possible within Canada has been the result of fixing the GM-GIA gene into all existing lines. The fixation of this gene, however, had reduced genetic diversity as a result of this continuous crossbreeding to make sure that every single plant had that gene. The biggest loss in genetic diversity was the result of the fixing of this GM-GIA gene, which was actually necessary to make growing soybeans in North America possible. To make a long story short, this gene and its many derivations control flowering time in soybeans. All University of Guelph lines have fixed this gene for one particular derivation or allele, and all Rajcan lines have fixed it for another allele that's suited to their environment. But in spite of this fixation of traits, the gradual incorporation of more diverse lines has led to the development of greater genetic diversity, and it's only increasing from here. And it is possible that the genetic diversity lost over the years could be. Another paper by Dr. Rajcan, this time solo, has tried to figure out the impact of OAC Bayfield in the breeding programs. OAC Bayfield is a crossbreed of multiple different cultivars. Full lineage of this plant can be seen on screen right now. OAC Bayfield has likewise been used to breed a great many different cultivars, both for Ridgetown and for the University of Guelph. And prior to that, approximately 65% of all genes in the Canadian germplasm used in making soybean came from 5 cultivars: Lincoln, which made up 24% of the common genome for northern varieties, Mandarin 17%, Richland 11%, AK Hero 7%, and Mukden making 5%. The fact that OAC Bayfield is becoming a bit of a stud plant in the breeding of new varieties has led to the question, how will this affect genetic diversity even further? And here are the results. It is revealed that the increasing use of Mandarin, AK Hero, Richland, and Lincoln has actually increased the amount of genes that have shown up. Mandarin genes have been found far more predominantly within Hero soy than its other parent. Similarly, AK Hero and Mandarin are on both sides of the pedigree, and the alleles from these genotypes have been overexpressed within certain lines within OAC Bayfield. This is likely due to selection for disease resistance, of which Mandarin is known for. This indicates that genetic diversity is not an end in itself, and genetic diversity that leads to more susceptible plants is not worth selecting for. Another aspect would be the impact of FISCB5 in OAC Bayfield's pedigree through its use in crossbreeding. FISCB5 was a cultivar bred in Sweden by Dr. Seven A. Holmberg in 1950 for the environment of FISCB, which is far more northern than most soybeans are well adapted for at that time. Based on genetic analysis, a great deal of the traits found within Bicentennial, the second direct parent of OAC Bayfield, still held most of the favorable genes that made it suitable for Russia, Sweden, and Ukraine. And that about covers every. Thank you for watching this video. If you enjoyed this video, please like and subscribe. If you really enjoyed it and think you can donate, you can do so at Buy Me a Coffee, link in the description below. Thank you for watching.